Hi, I am Mrs. Sloan, and this video is intended for my honors biology students. This is the first video in our new unit, Unit 6. And in Unit 6, this is called the Central Dogma Unit. So we're going to talk about, in Chapter 24, we're going to be talking about DNA structure. We're going to be talking about transcription, translation, and biotechnology. And then in Chapter 25, we're going to be talking about control of gene expression, which genes are turned on when. And we have already discussed in my class development, so we're going to get into that cellular specialization because certain genes are turned on. So that's the big overview, but for this video today, it's just about DNA structure and replication. So if you haven't watched one of my videos before, the notes are down in the descriptors. And additionally, there is a link to this presentation I'm giving right now. If that would be helpful for you, then you will have that video available, or the Google Slides available for you as well. All right, so in Chapter 24, we're going to do 1, 2, and 3, and today is DNA structure and replication, and then like I said, transcription and translation and biotech will all be under Chapter 24. So the first thing we want to go over is the big ticket items that you know, and this information though was not always known, and so I want to go into the background of some of the key experiments to help us understand about DNA being our genetic code. So here you can see a chromosome, and you know chromosomes are made out of DNA, and you know that we can use this DNA to build proteins, and that'll be part of this chapter as well. So DNA only has 4 bases. Can you remember what those 4 bases are? Did you say adenine, thymine, guanine, and cytosine? Whereas we have 20 different amino acids. And do you know why we have 20 different amino acids? What's different about them? Did you tell me the R groups? Yay. So when science was looking, that they knew inside the nucleus that there was both DNA and proteins, you can see, like, if you were trying to write poetry with 4 words versus 20 words, you would think, I could write better poems with 20 words. And so you would think that it would be proteins that would be our controlling information, but no, it's DNA. And that's because there's so much of it, we can have a lot of variety. So just a reminder, here are the 4 bases that we have in DNA. And here are 20 different amino acids that we have in protein. So there were several experiments to decide, so what controls the traits, the DNA or the proteins? So the first experiment we're going to go over is with Hershey and Chase, and they worked with a bacteriophage. Now, a bacteriophage is just a virus that only infects bacteria, and they look like a lunar landing module right here. And they did not know this because they did not have the microscopy to see what a bacteriophage was. All they knew for sure is that a bacteriophage, or a virus, consisted of 2 things: protein and nucleic acid, but they didn't know what was where. Now we know that the nucleic acid is encapsulated within a protein capsid. So here you can see in this picture, what you see here is the capsid inside, and I need a pointer, hang on here. Inside you can see this nucleic acid. They didn't know that was the arrangement, but what they did know, besides what a virus is made out of, is that it could overtake a cell. And what did it overtake it with? Did it overtake it with protein, or did they overtake it with a nucleic acid? So they did this experiment where they grew up viruses with some where the nucleic acid was labeled and some where the protein is labeled. One of the 20 amino acids that we have is cysteine. That's spelled with a C-Y-S-tine. But it has sulfur in it. And so they used an isotope of sulfur that they could, that would act as a tag, so they could follow wherever the protein went. You could trace it because it would be S35. The nucleic acid was tagged with a radioactive phosphorus, P32. So wherever the DNA went, they could trace it because it was radioactive, right? And so what they were hoping to find, they knew they had built viruses, and they knew viruses had proteins and nucleic acid, but what was it putting into the bacteria? So they needed to follow the tag. Did it put in radioactive sulfur? Was it a protein? Or did it put in radioactive nucleic acid? Was that this phosphorus that it put in? So here you can see this experiment when they had viruses, and you can see the nucleic acid is just colored yellow for simplicity in explaining this. And so when they allowed those bacteriophages enough time to infect the bacteria, and these phages are not drawn to scale, they're way, way too big. Okay, they would allow it to infect, and then they would want to separate the virus from the bacteria. So they put it in a blender, I kid you not, and it would separate the viruses from the bacteria. And then they would centrifuge. And because the bacteria is so much larger, it would go to the bottom of the centrifuge. And sure enough, all the radioactive phosphorus was at the bottom. So that gave evidence that what the virus was giving to the bacterium was its nucleic acid, since the nucleic acid was labeled with radioactive phosphorus. But let's double check, and let's do the same experiment, but let's label the protein. And so here you can see the capsid is kind of glowing. They gave enough time, put it in a blender, separated it, and you know what? All the radioactive stuff was on top. It was that radioactive sulfur was still with the virus, because that is not what it injected in. So on your notes for Hershey and Martha Chase, this is 24.1 in the notes, worked with a bacteriophage, a virus with a nucleic acid core and a protein coat, which is what we know now. They didn't know that then. And the phage would inject its blueprint into the bacterial cells to make more phages. Radioactively labeled proteins with S35 and DNA with P32 was discovered only. The P32 inside the bacteria. The only thing discovered inside the bacteria was the P32. The conclusion is DNA is the genetic material that is altering the bacteria. DNA is the genetic material that was altering the bacteria. So that's one classic experiment. And mind you, I'm not giving them all to you. Another one was done by a man named Chargaff. Now, whenever these guys win awards, they're always these old men. And I just wanted you to see what he looked like as a young man, as a young scientist. Lived from 1905 to 2002. And what he then analyzed were the chemical contents of the DNA, specifically the bases, okay, which we know are adenine, thymine, guanine, and cytosine. Now, because we've already studied the 4 important organic molecules, you know adenine is a purine. It has 2 rings, and it's always bound in a hydrogen bond with thymine in DNA, and guanine always binds with cytosine. Now, they didn't know this yet, right? These are these discovery experiments. So when he did his experiments, he had 2 things that he found out. The composition of DNA would vary from species to species. Different species had different amounts of these bases, but in each species, whatever percentage adenines it had the same percentage of thymines, and whatever percentage of guanine, it had the same percentage of cytosine. Okay, so here you can see several different species. They don't have, like, look right down here at just the adenines. They all don't have the same amount of adenines, right? But whatever percentage of adenines they have, in that species, they have the same percentage of thymines. Now, he did not know what to do with that yet. This was just one piece of evidence that helped with understanding the structure. So for Chargaff, and this is in the 1940s, the amount of A, T, G, and C varies between species. However, in each species, the A percentage equals the T percentage, and the G percentage equals the C. So A's and T's are equal, and G's and C's are equal. So here you can see that in this graph, A's and T's and G's and C's are about the same. So riddle me this. If I told you, okay, if I told you that 20% of the bases in a DNA sample were adenine, could you tell me the percentage of the rest of the bases? Could you tell me that? Hit pause if you want to, work it out on a piece of paper. Okay, you ready? Okay, so what does adenine bind with, right? Adenine, you know, binds with thymine. So if adenine is 20%, then thymine must be 20%, right? So that's a total of 40%. So what's left in the DNA? 60% is left over, and that 60% would need to be split between guanine and cytosine. So you just need to know the percentage of one of the nucleotides, one type of nucleotides, and you could tell me all of the rest. All right, so this was another experiment. And then you had Rosalind Franklin. Now, Rosalind Franklin and Maurice Wilkins worked together, but not very well, and she ended up going off on her own. She made some pretty critical discoveries which helped, and she may have willing or unwillingly given that information to Watson and Crick, and they ended up getting a Nobel Prize for discovering the structure of DNA. And she would have gotten it too, but she passed away due to cancer, and so they don't give those awards to people that are not alive, the Nobel Prizes. So she never got one, but she was a major contributor in this process. And let me tell you what she did. She would take crystallized DNA and x-ray it, just like if you broke your leg and you got it x-rayed, and it made this pattern right here of an x-ray. And from this, they knew that it was a double helix. She knew, deduced that that DNA had to be a double helix, that it had to be of constant diameter, and that there were repeating units. That's what she could tell. All right, so on your notes, for Rosalind Franklin, produced X-ray diffraction photographs and showed that DNA is a helix, constant diameter, and has repeating units. Okay, so now Watson and Crick have Chargaff's material, his data. They have Rosalind Franklin's material. They know the chemical components of DNA. They know it's a base, sugar, and phosphates, and they understand bond angles. And so they were able to put together the actual structure of DNA. And this is when they were younger, when they discovered this, and this is when they were older. And so they came up with this idea of a double helix. And here you can see their original model that they made. And then here's Rosalind Franklin right here, because she contributed so greatly to that. So what they discovered is, and I gave you this in the notes, in 1953, worked out the structure of DNA using Chargaff and Franklin's data, and they got the Nobel Prize in 1962. And the structure, a twisting double helix ladder made of 2 strands of nucleotides held together by, and do you remember, hydrogen bonds, held together by hydrogen bonds. And the sides are alternating phosphates and sugars. Okay, the sides are alternating phosphates and sugar backbone. And then the rungs of the ladder, you have paired bases. A purine is always with a pyrimidine, right? Adenine and thymine are always together with 2 hydrogen bonds, and guanine and cytosine are always together with 3 hydrogen bonds. And then the sides of the ladder, if you imagine this as a ladder, right? The sides are phosphate and sugar. The rungs of the ladder would be those nitrogenous bases. So let's look a little bit more at that. And for my students, this should be a review. I just wanted to show you here the diameter, 2 nanometers here across, a complete turn, like all the way around. If you were going down a spiral staircase and you were facing the front again, is 3.4 nanometers, and it's 0.34 nanometers between each rung of those bases. All right, and a little bit more on the detail. Remember, nucleotides are a nitrogenous base, and then a 5-carbon sugar, and then a phosphate group. Now remember, DNA has adenine, thymine, guanine, and cytosine, but we need to remember which one of those is not found in RNA. That's another nucleic acid. Do you remember? Right? You don't have thymine. Instead, you have uracil, little Dora the Explorer here. DNA, this is deoxyribose, but RNA is ribose, right? And the phosphate groups are the same. So here you can see the 2 sugars. This should be a review. Here is ribose. Look, OH, OH down here at the bottom of the house. And here is deoxyribose. Notice this would just be a hydrogen right here. I want to draw attention again to the numbering on the carbons. So carbon number 1 is the carbon that is connected to the nitrogenous base. And then you can't go up on the roof if this is a house because there's an oxygen here. So you go down 2, 3, 4, and the chimney makes 5. And it's the same thing for DNA. And that's going to become important in a little bit. All right, and then this is showing you the base pairs. They're complementary, adenine, thymine, and guanine and cytosine. Purines, 2 rings, pyrimidines are 1. I wish I could draw a line right here because between guanine and cytosine, there are 3 hydrogen bonds. Now, here you can see the 2 chains, but what I want you to see right here is look at the sugars, how they look at like houses on the left. And you see on the right how the houses are upside down. That's because they are anti-parallel. So it's kind of like a street and the traffic goes in 2 different directions. So here's carbon 1, 2, 3, 4. I go up here on the chimney, makes 5. So if I have an eyeball right here, I'm looking at the 5' end. This is what's called directionality. Just like we read from left to right, that's the direction we read. DNA runs. There's two different ways. See, this would be the 3' direction because this is carbon 1, 2, 3. So it's running this direction. And then when you look on the other way, look right here at the top. Here's carbon 1 connected to the nitrogenous base. We can't go this way because this is where oxygen is. Remember, at the point of the house, that's where oxygen is. So 2, this is the 3' end, and then this is the 5'. So it's running in two different directions. That becomes really important with DNA replication, right? And then this is just another picture to show you where the hydrogen bonds are. Okay, and you can see there's 2 hydrogen bonds between adenine and thymine, and 3. And because you always have a purine with a pyrimidine, for rungs, that keeps the diameter the same, that 2 nanometers across. You can see the phosphates. And this is called a phosphodiester bond. Here's a phosphate, and where it's connected to this sugar and this sugar, those are phosphodiester bonds. All right, so hopefully that is a good review of DNA. The next thing I want to talk to you about, I'm going to show you one more picture. Take a look. Nothing should be, look at the way this one is going and the way the carbon's numbered. 1, 2, 3, 3' end. You look across the street here, 1, 2, 3, 4, 5' end. If you're up here and you're looking down, okay, I'm looking at the 3' end here and I'm looking at the 5'. If I go down here and I'm looking up and I'm looking up over here, I'm looking at a 5' end, and over here, right there, I was looking at a 3' end. Okay, now if you see a picture like this, could you label this? I'm going to have you try this in class to try to label this with this information. Could you do that? And if you can, you might take a screenshot of it. And if you have an app like Notability or something, see if you can do that, because I am confident from what we've talked about that you could label that diagram. All right, let's talk about DNA replication. So your DNA that you have basically unwinds, unzips, and each of the original strands serves as a template for a new strand. That is called semi-conservative replication. So here's your original strand in blue. It unwinds and unzips, and each of the original strands serves as a template. Basically, the enzyme DNA polymerase needs to bring in the complementary base. So if the original strand here in blue had adenine, then it would need to bring in a nucleotide that contained thymine, right? If it had guanine, it would need to bring in a nucleotide that contained cytosine. And that's called semi-conservative because each has one old strand and one new strand. So on your notes, replication of DNA, semi-conservative replication, each daughter DNA double helix contains an old strand from the parental DNA double helix and a new strand, an old strand and a new strand. Okay, and you can see that in this picture, original in purple, new strand in green here, new nucleotides came in. Okay, and I'm going to introduce some enzymes. I want you to know there's more than what I'm presenting. If you're in my AP class, I would have you learn a little bit more. I'm going to limit the number of enzymes, but you might see them on a few pictures, but I'll help you to know which ones. So first of all, there's an enzyme called helicase, which helps the DNA unwind and unzip. But as it's unwinding one part, it kind of super coils it right where it's starting to unwind, kind of gets knotted up a little bit tighter. And so there's something called topoisomerase that helps. So both of those, helicase and topoisomerase, helicase does the major unwinding to expose those nucleotides. DNA polymerase, there's a couple of those. There's DNA polymerase 3 and DNA polymerase 1. I'm just going to tell you DNA polymerase is what's bringing in the new nucleotides. And because there has to be some primer that you put down, it involves ligase to seal the phosphate sugar backbone. So on your notes, number 2, underneath replication of DNA, the old strand serves as a template for the new strand. 2 strands are held together by hydrogen bonds. And then I'll go over those enzymes with you and show you a few more pictures. Okay, so here you have helicase, which is unwinding and unzipping this DNA. And then this kind of gets, when it's unwinding, this kind of gets coiled a little bit tighter. And that's why I mentioned that topoisomerase. There is a leading and a lagging strand. Now, remember how I told you they run in 2 different directions? Well, these enzymes, they read the DNA from the 3' end and they make 5 to 3. DNA is always made in the same direction, so is RNA, in the 5 to 3' direction. So they have one strand that it can go really quickly on the leading strand, and the other strand they have to keep backing up and turning around and backing up and turning around and making the DNA in fragments called Okazaki fragments. So that lagging strand takes a bit longer in order to do that. Okay, now if you start in the middle of the chromosome, remember they're like this, right? So if you start and you're going to replicate this top strand right here, in one direction it would be leading and you just get to take off. In the other direction it would be lagging. So you have to turn around, come back, turn around and come back. Notice it's all going in one direction to this DNA strand. Look across the street where the traffic's running the other direction, then they've got to copy it this way. So kitty corner from that leading strand is another leading strand, and then turn around, come back, turn around and come back. These areas, as they're working out, looks like a fork in the road. This is called a replication fork, and oftentimes they just show you the left-hand side of this picture, but you'll know it's going in both directions. All right, so here we're seeing that replication fork, okay, and they can read the 3' end, no problem. DNA polymerase takes off, zippity-doo-dah, bringing in free nucleotides, the original blue strand serving as a template. All right, but across the street, this one's a little bit slower. I have to back up, turn around, back up, turn around. And that's why you need this DNA ligase, is to seal all the little fragments together. All right, so let's go over our enzymes. I got them all spelled out here for you, and that goes in your notes as well. DNA helicase unwinds and unzips the DNA strands from each other, unwinds and unzips the DNA strands from one another. DNA polymerase positions the new complementary nucleotides to each other on the original strand. And then DNA ligase seals the phosphate sugar backbone, and making those phosphodiester bond. And the results are 2 double helix strands are identical to each other, and the original parent strand. And you know what those are? They are sister chromatids that are going to separate, right? Remember, sister chromatids held together by a centromere? And when you do mitosis, you can separate those sister chromatids from each other, right? That's where they came from. And when did we make it? In the S stage of the cell cycle. Okay, that's it for video 1. Video 2, we're going to talk about how we use this DNA to ultimately build proteins. And if you're one of my students, I'll see you in class.